Welcome, science friends, to Intro to Mutations. So a really quick review about the central dogma of molecular biology. This is really important and it applies to mutations as well. DNA is transcribed into RNA. RNA is then translated into protein. A gene is a segment of DNA that codes for a protein. So how do genes get the code for a protein? Well, that series of nucleotides. The transcription process from DNA into mRNA. Then that mRNA is going into a ribosome. It is working with the tRNA as well to create the protein or a chain of amino acids. A lot of times when we think about mutations, we think that, oh, they're really bad and they're going to hurt us. But not all mutations are harmful. Here are some examples of non-harmful mutations. We're going to look at eyes and eye color. So we're looking at 3 different examples of something called heterochromia. Hetero means different, so we're looking at something that's different, and chromia means color. So we're looking at different colors, perhaps within the same person. All of these women are fairly famous actresses, and we're going to start on the left with Mila Kunis. She has what's called complete heterochromia. Each of her eyes is a different color. One of them is more blue, the other is more brown. In the middle is Kate Bosworth. She has what's called sectoral heterochromia. So there's a section of her eye that is different than the rest of her eye color. One of her eyes has a section of brown, while the rest of it is blue, and then her other eye is completely blue. The last example is Angelina Jolie. She has what's called central heterochromia. So central means the center. The very center of her eye is a different color than the outside of her eye. So she has sort of rings of different colors in her eye. Very interesting. These don't hurt the women. It's just more of a interesting attribute to how they look. Another real-world example of a mutation that we're going to take a look at in the next video has to do with hemoglobin. Hemoglobin is a protein found in red blood cells, and it's about 200 to 300 amino acids long, so it's a fairly long chain of those amino acids. Hemoglobin helps our red blood cells trap oxygen. Here in the top right corner, we see a normal, healthy blood cell. It's very round and plump, and sometimes there's a mutation that occurs that changes the shape of our red blood cells. A mutation is a change in the nucleotide sequence of a gene. So when we're talking about hemoglobin, it only takes one nucleotide to change, and then the whole protein structure is going to change the shape of a red blood cell. So on the left, we see again that normal, very round, plump red blood cell. And when we just change one of those nucleotides in this one codon, CTT, to a CAT, that change creates a difference in amino acids. We go from glutamine to valine. And that change in amino acids goes from that nice round plump red blood cell to what's called a sickle cell. And on the right, you can see that the sickle cell is a sort of crescent shape. It's sort of hooked instead of that nice round shape that we want to see in a normal red blood cell. This is a harmful mutation, and it happens in specific populations of people when we see that change in a single nucleotide. When we talk about mutations, there are 2 big categories. The first is called chromosomal mutations, where there's a change in the number of chromosomes or the structure of those chromosomes. So sometimes we can have too many or too few chromosomes. Most of the time that's pretty fatal, so the baby will not live. But sometimes it's totally fine and that baby will live, probably with some impairments. There can also be a change in structure of our chromosomes. So sometimes pieces of our chromosomes can just break off, disappear, or change location, which isn't great for that organism. The other type of large category of mutations is called gene mutations, and that's the one we looked at with our red blood cell, where there's a change in a few of the nucleotides on a gene, or it could be just one, like we saw with the sickle cell. Chromosomal mutations, again, it's the change in number or structure of a mutation of a chromosome, and there are 4 main types. There's deletion, where we lose part or all of a chromosome; duplication, where a piece of it is repeated; inversion, where something is mixed around and changed and reversed; or translocation. And translocation is breaking off a piece and going somewhere else, to sometimes even a different chromosome. Here we see some examples. Again, deletion, we lost the B section of our chromosome. For duplication, we added a second B, so we duplicated that B section of our chromosome. Inversion, we see that things are mixing up inside that same chromosome. So instead of A, B, C, D, E, F, our sections got mixed around. Now it's A, E, D, C, B, F, so it's all mixed up. Translocation is when we're swapping pieces between two different chromosomes. So here, instead of A, B, C, D, E, F on one, G, H, I, J, K, L on the second, they've mixed up pieces. Now it's A, B, C, J, K, L, G, H, I, D, E, F. So things are all sorts of mixed up between different chromosomes. We're not really going to focus on chromosomal mutations. We're going to focus on the next kind, the gene mutations. So we're looking really at the few nucleotides, or sometimes even just one nucleotide in a gene that changes, causing something to happen with a protein. There are two main types, point mutations, and we're going to be looking at substitution. So we're swapping out nucleotides to create a different code. And we're also going to look at frame shift mutations, where sometimes one nucleotide leaves or comes in, we delete it or we add it, or sometimes we can even do that with 3 nucleotides, one codon at a time. So we could bring in an extra codon, or we can delete a codon, and something crazy happens with how that protein gets made. Some important vocab for us to learn. Wild type. Whenever you see wild type, that just means the normal sequence, the way it should be. Point mutations are just a change to a base. So something, it should be an A and it gets changed. It should be a G and it gets changed. So just one single point on that gene gets changed. And we're going to look most closely at a substitution point mutation. So a substitution means we're swapping out or swapping in. Something is coming in and out where it should be more like the wild type. So for example, maybe we put a U instead of an A if the A is supposed to be there on the normal sequence or the wild type. But then we have a substitution for that A, we get a U instead. We call this a point mutation because we're only changing one single base at one point in our gene. That was Intro to Mutations. I hope it helped.